may not think a whole lot about oxygen in your daily life. Hopefully you don't. But you would if you were up in the top of Mount Everest like this fellow is. There's about 30% of the volume of oxygen there as we have where we're standing now. And humans actually can't survive at that level of oxygen. We need about a minimum of about 70% of what we have in the atmosphere to be able to survive well. So oxygen matters. It matters to us. It matters to all animals in various ways. And just to look into the oceans, we can look at a variety of different organisms in the oceans that are also impacted by oxygen. This chart gives the minimum levels of oxygen that are possible for these different organisms to live. If 9 milligrams per liter is air saturation, the striped bass at 6 milligrams per liter has a similar oxygen requirement that we do. You can see that most of the fish actually have some reasonable requirement for oxygen, but some don't. Some organisms, like the bristleworm down here, for example, has a relatively low requirement for oxygen. So animals, as a spectrum of organisms, have a very broad requirement for their oxygen levels. And this turns out to be extremely interesting because the oxygen level through Earth history has not always been what it is today. If we look at this graph, this is something that I put together for that book that Carl mentioned called Oxygen. It's sort of a best summary of the available evidence on the evolution of oxygen through time. And we're pretty certain that oxygen levels have varied by at least 6 orders of magnitude over the course of Earth history. Oxygen levels are extremely low in the early part of Earth history. There was a period of time around 2.5 billion years ago, 2.4 billion years ago, where oxygen levels rose dramatically, probably about 3 or 4 orders of magnitude. This may or may not have been association with the evolution of cyanobacteria, which are believed to evolve sometime around here. And for those that don't know it, cyanobacteria were the first organisms on Earth to produce oxygen. Then oxygen was much higher through what we call the Earth's Middle Ages at levels that are somewhat uncertain. We'll get back to that just a little bit in the talk as we go on, but certainly higher than they were before. And then we come to the Phanerozoic, which is an interesting period of time when we start talking about here, because the beginning of the Phanerozoic is when animals actually evolved, or at least large animals started to evolve. The animals themselves started to evolve probably sometime around here. And when we get back to that original slide, or the second slide that I showed, there's a discussion about whether or not the timing of animal evolution was also related to timing of oxygen change or increases in the atmosphere. And that's a point that we're also going to get back to a little bit later on in the talk. So this is a broad history of the concentration of oxygen. One of the thoughts that I want you to try to keep in mind as we go along is whether or not this history of oxygen has impacted the evolution of animal life. But sort of in the spirit of Kor's excellent talk and Sergei's excellent talk recently, I'm going to try to keep the details down and to try to keep the explanations up so you can see a little bit, hopefully, as to how we understand something about the oxygen history and how we understand something about how this might have impacted the evolution of animals. So it all starts with the rock cycle. And I show this as sort of an element of basic geology, but maybe I should use this here, pointer thingy. But also because it demonstrates a couple of points that I want to make. One is that materials are weathered at the Earth's surface, and that means that they are impacted by dissolution and abrasion and processes like that, and they're disaggregated. So you take a stone, a rock at the Earth's surface, over some period of time, that stone is going to become weathered, disaggregated, and transported into the oceans. And it's transported in 2 ways. It's transported either as this disaggregated solid material, or some of it becomes solubilized during the weathering process. So we have this transport of what was rocks at the Earth's surface into components which are both dissolved and solid into the oceans. And these components deposit into the oceans, and then some of them are uplifted later on back to form land again, and then to keep the cycle continuing. Some of them are also subducted back down into the interior of the Earth, which is also very interesting. But the point is that we have a rock cycle, and this rock cycle delivers substances to the oceans, and it's those substances we're going to be interested in. In fact, lucky for us, some of these substances are impacted by the amount of oxygen in the atmosphere. So, for example, organic carbon. We'll get back to this organic matter. Organic matter can be sequestered into rocks sequestered into sediments as the sediments form that become rocks. And this organic matter then is weathered again at the surface, and the amount of organic matter that gets weathered depends on the amount of oxygen in the atmosphere. Other minerals like pyrite or molybdenite or uraninite, their weathering is also impacted by the amount of oxygen in the atmosphere. And what that means is with there's sufficient oxygen, then these compounds can be solubilized into these compounds, and these compounds then are freely distributed into the oceans after they are transported there from rivers. Now comes the fun part. We're going to look at a particular element in particular. We're going to focus on molybdenum, so you get some sense for how this might work. You know, how we sort of try to reconstruct something about the history of oxygen. So, on land, molybdenum is found in a variety of places, but a black shale, a black rock, which used to be sediment, is a typical place to find it as molybdenite, MOS2. The molybdenite reacts with oxygen through what I'm going to call just oxidative weathering and forms molybdate ions, which are then fluxed into the oceans. Now, what's important is how molybdenum comes back out of the oceans again. It doesn't just accumulate, it's in some kind of a steady balance, so some of it comes back out again too, of course. So some of this molybdate adsorbs onto manganese and iron oxide particles, which you can find in the deep ocean. A very strong removal pathway is the deposition into anoxic water, sediments that are forming in anoxic water. And if you want to think locally about that, think about Maya Fjord, where the sulfide accumulates up in the bottom waters of Maya Fjord. If you want to think more distantly, think about the Black Sea or the Kariako Basin, or a number of fjords around the Norwegian coast. But this is a strong removal pathway. And then just sort of in typical muds like you find off the coast of Denmark and in all coastal countries, you have sort of a weak removal pathway. Now, let's try to put some logic here together. The first piece of the logic is that the flux of molybdenum to the oceans will depend on the concentrations of atmospheric oxygen. Probably not in a linear fashion like this, but that doesn't matter. Just keep it in your mind that it depends on the amount of oxygen, whether or not we get molybdate into the oceans. Okay? The amount of molybdate that we have in the oceans depends on the amount of anoxic water, or the strength of this very strong removal pathway. So you can imagine that if this anoxic water is widely spread over the oceans, this removal pathway is extremely strong, and the concentration of molybdate then should be low in the oceans. If there isn't much anoxic water in the oceans, then that strong removal pathway is not that well expressed, and the concentration of molybdate will be higher in the oceans. So we have an inverse relationship with the amount of anoxic water. Okay? Now, we come back to the atmosphere again, because the amount of anoxic water that we have in the oceans depends to some extent on the amount of oxygen in the atmosphere. The more oxygen in the atmosphere, the more oxygenated the oceans. The less oxygen in the atmosphere, the less oxygenated the oceans. So the more anoxic waters. Now, the last piece of the puzzle. How are we going to get back to something that we can actually practically analyze in order to know something about the extent of anoxic water and then atmospheric oxygen? So the amount of seawater molybdate you can estimate by the amount of molybdenum that you can analyze or find in a rock. So it comes out in anoxic waters in some proportion to its concentration in seawater. So we analyze a rock, We can estimate the concentration of seawater molybdenum. We can get the amount of anoxic water, and from there we can get some estimate for the amount of atmospheric oxygen. Right? It ain't that easy. That's a takeaway message. So how do we do this? Well, we go to the field, and this is a beautiful place in North China. It's called the Jiaomeling Formation with some absolutely spectacular sedimentary rocks. And for those that are interested in climate change, these bands are controlled by variability in climate during this deposition of this rock. We collect some samples of the rock, and this rock will contain these things: organic carbon, pyrite, molybdenum sulfide, uranium. We measure the concentrations of these. And then we do it in a lot of different places. And we get a record of the concentration of molybdenum over time in these rocks. And it kind of fits with our expectations now based on what we've been talking about already. There's very little molybdenum in rocks up until about 2.4 billion years ago. There wasn't oxygen in the atmosphere back then, so the oxidative weathering wasn't happening. Molybdenum wasn't being transported into the oceans. Here at about 2.3 or so billion years after the oxygen started to increase, we see an increase in the concentrations of molybdenum in rocks. Not as high as today. Okay, molybdenum was being transported into the oceans, but the amount of anoxic water was likely rather large, so oxygen levels were likely much lower. Now when we get further on in Earth history, around that time when animals started to evolve, the molybdenum concentration increases, implying that the oxygen content of the oceans was greater, the anoxic waters were less, implying again that atmospheric oxygen concentrations were higher. Okay, so it's a long string of logical events. Turning all of this into some real estimate of atmospheric oxygen, though, is very difficult. And I want to make a little plug for something that I'm very excited about, and that is that a lot of our actually quantitative translation of these processes I've been talking about into estimates of oxygen are based on a very, very limited understanding of how these metals are actually removed in modern sediments. So if you can see colors here, the purple colors here, it's about altogether about 50 or so different sites is where we have measured, not we necessarily, but but the field has measured molybdenum concentrations and has some sense for how these various pathways work. We've just recently gone and from the collection at Lamont Doherty Geological Observatory collected 50 or 60 more samples just to get a broader sense of how the bigger ocean is also responsible for removing not just molybdenum but other elements as well. And when you look at this map, we're looking at about 100 or 150 sites. The ocean is huge. So I'm saying that the ocean is so way underrepresented. We really have a very, very little understanding of the chemistry of the sea bottom. And this is something that I'm passionate about and I want to do something about. But it's a little bit of a side from the story, but just to show you that there are various elements of the puzzle that you have to put together if you want to get an oxygen estimate, and this is one where we're really lacking, I think, in our understanding. Okay, editorial over. Let's move on. So I'm just going to give you a brief introduction into another way that we're trying to analyze, isolate, understand the history of atmospheric oxygen. It's back to this organic carbon thing that I was talking about. So organic carbon, again, is found in rocks, and those rocks were once deposited or from the seafloor for sediment. They've been lifted back up onto the continent again, and now we have rocks on the continent. Oxygen reacts with the organic matter, generating CO2, but maybe some of this organic matter actually isn't completely oxidized and then is transported as this particulate material back into the ocean. So it's recycled carbon. And what's interesting is that the amount of recycled carbon that you find should depend on the atmospheric oxygen level. The more atmospheric oxygen you have, the less amount of the organic matter should be recycled, and the more carbon you have, or the more oxygen you have, the less should be recycled. So the question is then, can we figure out how much organic matter has been recycled through time? And thanks to my postdoc, Sammy, here, we can do that. We go back to the rock, and we use something called Raman spectroscopy, which is able to analyze the nature of carbon-to-carbon bonds in organic molecules, organic substances that are found in rocks. And what you find is that some of the organic matter is what we would call disordered organic matter, like this stuff here. And that's probably organic matter that was produced at the time the sediment was formed by primary production in the water column. It's the freshest, youngest stuff. But you can also find organic matter that has been graphitized. It's been heated to a higher temperature. It has a different history than this organic matter here. And you can even find some stuff that has been heated very high. This is clearly recycled organic matter. So we have organic matter that's not recycled and organic matter that has been recycled. So what we've done is we've gone through time. I won't drag you through all the details. We've gone through this Middle Ages part of geologic time that I mentioned. We've randomly looked at the distribution of this disordered and recycled organic matter in these rocks that we've looked at. We find that this stuff is very, very hard to find. Most of the organic matter is of this disordered nature, which means that the recycling of the organic matter was very efficient. And that has allowed us to put some estimates on atmospheric oxygen, which lie actually rather closely into some of the earlier estimates that we made. We have to model to do this, and I don't know how you guys, if you've done modeling before, maybe some of you have. What's the famous line about modeling? All models are wrong, some models are useful. So you always have to bear in mind that step from observation to actual estimates of oxygen requires some iterative in-between steps. That's the modeling part, and we want to try to get better at that, of course. But anyway, that's a part of the story. But now we come back to here. The question is, if this is a range for the oxygen levels during this period of Earth history, this is the range that we have now. There was likely an increase sometime around this period in Earth history. Did animals evolve in association with that increase? Another way of putting it is, what are the minimal requirements of oxygen for animals? And we're not talking about all animals now. We're talking about the earliest animals. So if we look at kind of a artistically generated animal phylogeny, and a phylogeny just means a history, you can see that we have the two main branches of animal life here, but before these branches emerge, we have a couple of forms of animals which represent sort of the beginnings of the animal evolution. Tree, and the ones that we're looking at are peripera, the sponges. So sponges represent early evolved animals. And there's geologic evidence for that too. Not a lot, but there's some. Here is an example of what was most probably a sponge from about 600 million years ago, with the main sort of features that you can identify in modern sponge, at least with respect to the architecture of the sponge body. So sponges are ancient organisms, and the question is then, how much oxygen do sponges need? And to do that, we landed on this fellow here, this Tethya wilhelma. And I don't know if anybody has ever tried. It's relatively easy to keep cats and dogs and snakes and stuff as pets, right? I mean, you know how to do that. Sponges are really, really hard to keep in captivity. I mean, they're simple animals, but it's hard to understand what they need and where it goes wrong. So it's actually really difficult to do experimental work on sponges because they die all the time. But these guys don't. The Tethya are actually reasonably well behaved. So it's actually for that and another reason, which I'll get to in a moment, that we chose to work on Tethya. So if you want to try to figure out how much oxygen Tethya needs, one thing you can do is just a simple experiment where you put Tethya into a tube and you look at the drawdown of oxygen, how it respires the oxygen. You look at it over time. And at some point, at some oxygen level, the oxygen concentration starts to impact the respiration rate of the Tethya. And at that level, you would say, okay, now oxygen concentrations are starting to impact the respiration of the organism. That happens at about 25% oxygen level. And then we often sort of fit this kind of data with something called a Michaelis-Menten curve. And if we do that, then we can calculate the half saturation of oxygen. And we find that the rate of oxygen uptake is half of its optimal rate at about 5% of atmospheric oxygen levels. So you can sort of imagine then that at least respiration starts to impact, or oxygen starts to impact Tethya respiration at around between 5 and 20% oxygen levels, something like that. But what does that mean? Does that mean that that's what Tethya requires? Well, we can look at it another way. And this is so cool. The other reason that we chose Tethya is because it, oh, come on now, let's be nice. It's because it does this. Can you see it? It's contracting. It has these regular, about every 4 hours, it has this regular contractile behavior. It's a behavior. The sponge has a very, very dedicated, determined, reproducible type of a behavior. All Tethya sponges do this. And we can look at it again. Really cool. So what we did is we tried to figure out how oxygen impacts this behavior of the sponge. So the logic being here, if the behavior of the sponge is being impacted by oxygen levels, then that has something to do about how the sponge, the sponge environment is impacting the sponge. So when we looked at this, it's kind of a messy graph, but the thing to concentrate on is this. This is the contractions per day, and Tethya went from about 2 to 3, let's say 3, contractions today at 4% oxygen levels down to no contractions at 2%. So we would say that the behavior of Tethya was impacted in this oxygen range, and I would argue that that also means that Tethya as such was affected by oxygen in that range there. Does that mean that Tethya needs that much oxygen? That's another question. And in order to look at that, we just took Tethya and we exposed it to very low levels of oxygen for an extended period of time, 2 weeks. So we have about 0.2% atmospheric oxygen. And this is, I mean, you know, you get a sense like when your kid is sick and not doing well, right? You get a sense if you have a dog and they can't really walk, or you know what I mean? You get a sense from when organisms aren't that happy. You know, how do you know when a sponge isn't happy? You know, it's not contracting anymore, but it's keeping its color. It's not decomposing it. The cells aren't obviously dying. So I would argue that this Tethya can at least survive under oxygen levels, which are below that 2 to 4% sort of minimum that we've looked at for where this behavior is impacted. So it doesn't need much oxygen. I think that would be the point. Does it need more than that? Maybe it does, but 2%, 3% seems to be enough. Now, another thing about sponges and oxygen, these are actually sponges. You probably can't see that easily. They're encrusting sponges. They're growing on rocks. And these are found in a saltwater lake in Ireland called Loch Hyne, which is daily inundated by the tide. And during the summertime, when the water is warm, Loch Hyne goes completely anoxic and sulfidic. So where these sponges live during the summer months is completely sulfidic. So they have 3 months a year where not only is there no oxygen around, but they're also being bathed in a solution of sulfide, and they think that's just fine. They survive this quite well, and we're trying to understand how they do that. I mean, you know, how do they do that? What sort of is the nature of the sponge body when it's exposed to hydrogen sulfide? You know, is it active in some way? Is it shut down its metabolism? You know, we've done some work, I don't want to get into the details, you know, too much here, that seems to suggest the sponge actually doesn't change very much from the oxygenated to sulfidic conditions, which is actually very, very surprising. But we need to learn more about this. But anyway, the point is that the sponges, some of them don't even need much oxygen, but they can survive anoxia also for at least some periods of time. So, okay, now to sort of wrap things up into the last part, there's another element of sponge oxygen which is really interesting, and it's related probably to evolution as well. But I want to show you this. This is the work of Lars Kumala. And what Lars is an expert at is growing sponges in the lab. So you can take a piece of Halichondria panacea, that's the normal breadcrumb sponge. You can find them all over the place. You can go out to Katamini, you know, you look down in the little porpoise viewing area, you can see these sponges all over the place. They're very, very, very common. But you can take a little, like half a centimeter cube of these sponges, and you put them on a slide, and then over some time, they grow into fine little sponges like this with a single osculum. That's the exit port here. So the sponge takes in water around its cell body in various places and spits it all out in the osculum here. Now what we do is we grow these sponges on slides that have a chemical dye, a fluorescent dye, that when excited with the right wavelength of light, fluoresces based on the oxygen concentration that it's being exposed to. So what we can do is we can get a two-dimensional picture of the foot of the sponge, the oxygen levels of the foot of the sponge. And here is an example of what that looks like. This is a time lapse over about 2 or 3 days, something like that. And you can see that the sponge periodically goes anoxic. This is kind of related to those contractions we saw in the tethia. It closes its osculum, stops pumping, and then the interior of the body goes anoxic. And it does this in a very sort of regular way. And if we were to do this for a week, it would keep doing it with the same pattern. It might not be that the regularity is exactly every 4 hours. It might be 4 or 6. It changes a little bit, but the same pattern of internal deoxygenation occurs. So the sponge has an absolute requirement to deoxygenate the interior of its body on a periodic basis. And we can analyze this. We can analyze the concentrations. We can look at the concentrations of oxygen in the sponge as they drop during the deoxygenation, during maximum deoxygenation, as they increase again. We can look to see what the average oxygen content is within the sponge, and we can look to see what percentage area of the sponge has different oxygen levels. I mean, this is just some of the analysis we can do on this. And we can also look at other species. So another species we looked at is Haliclona rosea, which is another sponge that you can find out in the Kattegat. And you can see this has a very, very different style of deoxygenation. It doesn't close its oscule in the same way that Halichondria does, and it has this really interesting kind of I call it the light show pattern of deoxygenation. This is a repeated image. We're not looking at a long series of time lapse. This is one event here, and this is another event over here. But this oxygen, low oxygen, is kind of transmitted through the body in a very interesting way. And if you want to get nerdy about it, it probably means that the sponge is internally regulating how oxygen is being sort of transported through its water canal system. So it's actively controlling its water canal system in order to be able to have this oxygen wave sort of flow through the body. But it's a messier system than Halichondria, so that sometimes deoxygenation is rather significant, like here and here, but many times it's not. You know, it's just sort of this wave just kind of rumbles through the sponge, and then it comes back to normal, complete oxygenation again. So, why do I show you this? I show you this because the real question is, why do they do it? I mean, why do sponges do this? Why do they do it? I should ask, if anybody has a really good idea, I'd like to hear it. We don't actually understand why sponges do this, even though it seems to be an absolutely critical part of their behavior. We have some hypotheses, though. One hypothesis, which I'm sort of moving away from now, is that sponges, like us, like all animals, have a microbiome associated with them. So one hypothesis would be that there's sort of crosstalk between this prokaryotic microbiome and the sponge itself, which is regulating the internal oxygen concentration. You know, if the microbiome is happy, then the sponge is happy. You know, that would be one way of looking at it. There's another way of looking at it, too. And that way has to be is more related to the sponge itself. Sponges don't have stem cells like we do, but they have cells that can transdifferentiate. They have cells that can change into other cell types. And one hypothesis would be that the sponge is periodically introducing low oxygen into the body in order to allow these sort of cells to transdifferentiate. In our own bodies, the stem cells do this under low oxygen conditions. So maybe there's an analogy to sponges too, that the sponge body, actually the sponge cells transdifferentiate under low oxygen levels. We don't know. We don't know the answer to that. But in principle, one could study it. And it might have some really interesting evolutionary sort of, I don't know if it's a consequence, but an evolutionary history anyway. Because if sponges evolved early on in low oxygen levels, they didn't need to be that high. I think we've sort of established that sponges could probably evolve in something like 2 to 4 to 5% atmospheric oxygen levels. And the geologic record suggests that amount of oxygen was around way before sponges evolved, which is another interesting point. But anyway, if the sponges evolved under those low oxygen levels, they may not needed to sort of develop this internal anoxia in order for them to be able to transdifferentiate their cells, in order for them to be able to survive. So if that was the case, and kind of an interesting development during evolution, as oxygen levels increased, the sponges reacted to that by actually developing a behavior which then allowed them to internally develop low oxygen conditions on a periodic basis. I mean, it's a little bit difficult to prove that, but I think it's sort of an interesting hypothesis into how animals might have had to cope with increasing oxygens. It could be that the sponge was actually better adapted to the low oxygen conditions and then had to cope in some way with oxygen levels as they increased. Food for thought. I don't have any answers here. Maybe you guys do. I'd love to hear about them. But I think I'll leave it there. And thanks for your attention.
Time for questions. So.
Hey Don, it is a fantastic talk here. I learned a lot from the point of the oxygen part of view here. So my question is regarding the oxygen, and you try to analyze it, in a different time and from the sedimentary rocket. And are you only analyze the one part of the rocket, or that is I guess you are from the rocket from the Shamalin, yeah? So. Because in the different time and the temperature and it's different, and there might be the other chemical reactions, and for example CO2 react with your MO and this element here. How can we make sure that the carbon, the organic carbon or the MO and the composition is exactly relevant to the oxygen level in the atmosphere? If there's other factor parameter inside, for example temperature and CO2 concentration in the atmosphere, how can we make sure that data?
That's an excellent question, and I think there are actually two parts to the question. The one part has to relate with, given that these rocks are representing what was originally there in the sediment, are there other factors than oxygen that could impact the amount of molybdenum, let's say, that's in the sediment? And absolutely, there is. So typically what we will do is we'll not just look at a single rock, we'll look at a whole sequence of rocks. And we'll not just look at molybdenum, we'll look at a variety of elements, and we'll try to put a story together based on a variety of different lines of evidence, not just one. But that's an excellent point. The other point is, how do we know that the conditions that rock deposited under haven't been compromised through time? Let's say if you're thinking about what happens, these rocks have been buried in the earth, right? So there are fluids that could flow through, and those fluids could impact the chemistry of the rock somehow, and that's a tougher one to deal with. And there we use geological context, where we look and we see, are there evidence for these fluids? Can we see them? If there is, we typically can see some telltale signs that they've been there and that they've impacted the chemistry of the rock, and we stay away from those rocks. Specifically with respect to the carbon, that's actually kind of interesting, because if we're looking at high-grade rocks as this recycled carbon, if this rock we're looking at is a high-grade rock, we can't get any information out of it. If itself has been heated up, right, to a level where this detrital particles have been heated up to, then we can't use it. So there are a certain type of rocks that we're restricted to analyzing for this. So yeah, compromises. Yeah. Oh, sorry.
So thank you very much. I'm coming from a completely different field from health, so maybe this is a really shitty shot at a hypothesis, but we know that reproduction is crucial for evolution, and I don't know anything about sponges, but just look them up, and they seem to be alternating between being male and female.
Yeah.
So could the oxygen differences in oxygen flow be depending on or determining their sexual state?
I have absolutely no idea, but that's an interesting thought. Note that down, Lars.
You have this suspicious dip in oxygen level at -2000.
Right.
Any explanation for that?
Yes. So this is a hop, skip, and a jump. Exactly. Let me go to there's a little bit of a logic train here. So it's so this initial rise in oxygen here, it weathered the rocks in a very different way. And one of the things that it would have done is it would have liberated more nutrients, phosphorus in particular, to the oceans. So for a period of time, on probably hundreds of millions of years, there was an enhanced load of phosphorus to the oceans. What that meant is the primary production got very high. And this is evidenced by the deposition of some very, very organic matter-rich rocks at about 2.2 billion years ago. Okay, now, these rocks start to become part of the rock cycle. They get buried, now they get transported back up into the zone of weathering again. Now you have all this organic matter that is now put back up into the weathering environment, and that's a tremendous drain on the oxygen content in the atmosphere. It's a tremendous sink for oxygen. So that's a hypothesis that we have like this sort of oscillation related to this carbon being transported back up. So the initial oxygenation, organic matter deposition, organic matter brought back up to the surface again, causing a dip. That was a good point.
Thank you very much for a wonderful talk, Don. I have two questions. One of them is about the ocean, where and the dots you were showing. How do you decide on the dots? That one, there's a lot of places you could go. And the second thing is about what you started out saying, that you were thinking about oxygen on a daily basis. People are actually also doing that in clinical medicine. It's a huge thing, from pediatric to geriatric. And like here, for sure, most of the focus is on too low oxygen. But actually, there's also concerns about too high, especially in infants and so on, where you can get blindness and so on. And you alluded a little bit to it in your final statement about there might also be too high things. So I was just wondering whether one should talk about intervals as optimal instead of lower limits only.
I agree with you completely, Cor, but let's take the second one first. Yes, intervals are a really good way of thinking about it. From an Earth history perspective, there was a time about 200 million years ago where oxygen levels were probably about 30 or 40% higher today. And have you ever heard about this giganticism in organisms? But there's also talk that we might have reached that limit where oxygen level actually might have been detrimental to life. So yes, for sure. My focus on the lower limit is mainly due to this hypothesis that we're testing. You know, whether or not animals were enabled, the evolution of the animal body plan. You know, the animal type of construction was enabled by an increase in oxygen. But yes, the idea of too much oxygen is a really interesting one. I agree. And that might have changed through time. That window of what was too much, you know, what present day levels might have been devastating to organisms 600 million years ago. Oh, and then the map. So the map, if we look at this map, most of the purple dots are these sediments which are depositing in low oxygen environments, because that's where we get a lot of our information from, if you remember that molybdenum example I gave. So there's been a very much of a focus on those types of environments. There's the Black Sea here, for example. Various zones of low oxygen sort of spread around in different places. So that's why these dots. Now this is just sort of a random shot, the blue dots in a distribution. My goal is to put 5,000 dots on the map. Because I think then we will really have a good understanding of what the chemistry of the ocean bottom actually is. That's my goal. That's what I really want to do. The blue dots, there's 54. We made them big so that they looked important. Think how big the ocean is, you know, that's nothing. It's really just nothing.
Is it possible to use citizen science for that?
No. But it's not possible to use citizen science for this, but it's possible to use citizen science for that. I didn't talk about it. So part of this project with Sammy and the carbon cycle, and there's recycling of organic matter to try to figure out what we have in rivers, because that's actually what's transporting stuff to the oceans. We were looking at ancient rocks, but what's happening in the modern world where we know how much oxygen there is, we have control over these factors that control it. Back when I was a Ph.D. student, the last part of my thesis, I packed a nitrogen cylinder in my truck and made a river trip through the continental United States and collected river particles from all over the place. And this it was really great. I read Travels with Charlie, and I don't know if you know these books, or Blue Highways by Least Heat Moon. I was having a real spiritual experience with this. But there are a few other rivers which have also been analyzed around the world, but not with the spectrum of elements that I'm interested in. So I want to use citizen science for that. I would love to be able to develop a package that I could send out to a high school student or anybody who was interested to go to a river, filter down some sample, and send it back to me so that we can analyze it. I need to think very hard about it. It has to be really bulletproof, you know, so that there's really easy to use. And one thing I worry a little bit about is whether it's dangerous. I mean, you know, people have to sort of assess that themselves, I guess. But you know, you put people to a river to collect water. You know, is there danger involved with them doing that? You know, I have to think a little bit about this. But
you do that already, don't you?
Yeah, but I don't put them into dangerous rivers, of course, right? No, I mean only in the coastal places. Yeah, but yes, we do. But I guess we can help you on this.
Yeah, but it's
what's dangerous. The Danish rivers are not so dangerous, though.
No. Well, but Denmark's a small place. We want to get some dots all over the place.
There's no dot in Denmark. No.
No, there isn't.
Just
You're
saying.
right. You're right. If I had a goal there, I'd like to get 200 rivers, and I would love to use citizen science to do it. And I have some ideas about how to develop a bullet fruit package, but I'm not there yet. So I want to do that.
All right, I guess we're there, right? So I'm just closing this day's talk and would like to give Don again a hand for this exciting talk and insights.